Welcome back. Up to now in the course, we've been talking about what science knows about cancer in terms first of the challenge of cancer, then some of the causes of cancer. And we found out first some of the causes from population studies and epidemiology, and then we studied DNA and how cancer and carcinogens act on DNA, and ended with some real dilemmas about how we evaluate cancer causes at the DNA level. Now I want to turn to cancer as a disease, and a disease at the cellular level. In an earlier lecture, I described the three main ideas of biological sciences, of life in general. First, mechanism. Cancer acts on DNA. Cancer is a disease of chemistry and physics. Second, cells. Biology is made of cells. Cancer is a cellular disease. Third, evolution, change through time by natural selection. Cancer, as you'll see, changes as a tumor develops. The story I want to use to begin this discussion of cancer in cells is the story of cell reproduction. Cell reproduction may be familiar to you. It's a series of events where chromosomes line up and get duplicated and then get separated into two new cells. It's really like a symphony. It's a complex series of events in which multiple players are acting, and it somehow comes together and ends up in a nice sequence of events. Well, just like a symphony has a conductor, there's got to be a conductor of events in the cell reproductive cycle. I'm going to describe two scientist teams working independently on different organisms that came to the same conclusion about the conductor of events in the cell cycle. The first scientist I'm going to describe is Leland Hartwell, who worked at the University of Washington in Seattle and is still active in research. He asked the question, what can we learn about normal cell division from cells that have a mutation, a genetic mutation in the cell division cycle? It is in that orchestrated series of events. Because in using this approach, you could compare the mutant organism with the normal organism and see what the mutant is missing, right? If the cell stopped at a certain point, what's missing that made it stop? The cell that he used was yeast. It's familiar to you in baking and other purposes. Yeast is a model organism, one of the model organisms biologists use to study biology. It undergoes cell division. It's complex. It's got a cell nucleus. It's got all the cell parts that a human cell has, really. Does it reflect us or other organisms? Yes, it does, because of our assumptions in evolution, change through time, that we all have a common ancestor. We all have the same chemistry. We all have DNA. We all express DNA in very similar ways, etc. The reproduction of yeast cells follows a familiar pattern. This is a slightly different drawing, photograph than the drawings I showed you earlier on. That is, a cell has a nucleus in it. The yeast cell forms a little bud as it begins to reproduce. So a little blip comes off of the yeast cell. The blip gets bigger and bigger. And in the meantime, the nucleus reproduces itself. It reproduces all of its DNA. And then you have two nuclei. And one of the two nuclei, as you see, migrates into the other bud. Hartwell isolated some mutants of yeast. And he found that one of the phenotypes of the mutants, that is the phenotype, the outward expression of the mutant, was that it was stopped in the cell cycle. So here's the cell cycle of yeast. And you'll see that instead of drawing it in a linear way or have two rows as I did, I'm drawing it now as a circle. And this is a cell cycle that's kind of universal in its characteristics. We start off with an individual cell, which here is called the mother cell. And then the cell forms a bud, you see that, and then the bud gets bigger, and now you have two cells at this point, and the cycle goes around again. Now we can divide the cycle into several parts. The initial part is the cell is kind of hanging out, doing regular things, but not duplicating its DNA. This is a cell, it's a typical, in a body, it might be a typical specialized cell. It's just there, it's acting and doing cell things. When a cell prepares to divide, it goes through a point called a start point in yeast. And after this point, the cell duplicates its DNA. This is called the S, or synthesis phase, and the DNA gets duplicated. It still looks like a normal cell, no compact chromosomes or anything. Then it goes through another period where it's just compact nuclei, etc. And finally goes through the phase called M, or mitosis, where the cell chromosomes actually appear. And I've shown you diagrams before and today of chromosomes dancing and migrating into two different cells. So this is a cell cycle typical of yeast or any other organism that's complex. Hartwell found a mutant that was missing something that allowed it to pass this point. In other words, the mutant stopped at the start point. It just stopped. It hung around as a cell. It should have been dividing, and it just stopped there. And so then he and other colleagues went looking for what was missing in that cell. What was missing in that mutant? Because that must be the thing that gets the cell to commit to duplicating its DNA. I said there were two groups of scientists. Here's the second group. Second group was James Mahler, working quite a piece away at the University of Colorado. Not necessarily knowing what Hartwell was doing, in fact. He tried an experimental approach, and he used a different organism. The organism he used is a popular one in biochemistry studies. It's the frog, and the frog is shown here, and it has a life cycle that many of you may be familiar with. It has an egg, and the egg is shed out into the water, and the egg makes a little polywog or a larva, and the polywog then forms a frog, and many of you probably are familiar with this. The egg develops inside the frog. Mahler studied the egg development of the frog. And what happens during egg development is the egg begins to develop, and then the frog forms multiple eggs. And when the immature egg is just sitting there, a hormone comes, it's made by the frog, knocks on the door of the egg cell and says, okay, get going. And then something happens inside the egg cell, some chemistry happens inside the egg cell, and the egg then gets stimulated to go into cell division. This is called the maturation promoting factor. It's a cell division promoter. It takes a cell that's just sitting there and stimulates it to divide. So Mahler decided to do an experiment, and here was the experiment he did. He took an immature egg cell of the frog in the laboratory dish, and then he stimulated with the hormone. And of course, the immature egg cell then did its thing. It began to divide. Okay, it stimulated itself to divide. Then he said, gee, there must be something inside that cell. So he sucked out the contents of the cell, of a stimulated cell. And he injected the contents into a cell that was not dividing. The cell had not seen a hormone at all. But he added the stimulus package, as they would say, to the interior of the other cell that was just sitting there. And that stimulus package then stimulated this other cell to go into cell division or mitosis. When Mahler isolated this stimulus for cell division, he found a very unusual, for that time, protein. And the protein was called a protein kinase. I'm going to repeat that, protein kinase, because this is a very important type of molecule in cells of many organisms. Here's the amazing thing. When Hartwell looked back at the yeast and asked, what's that mutant that doesn't go into cell division missing? You bet it was the protein kinase. So we have a universal thing. The same system that controls as the conductor for cell division in yeast is also the control cell division in frogs. And in later on, lots of other organisms were shown to have the same general mechanism, including humans, including cancer cells. What is this stimulator cell division? Here is the protein kinase. And I have to review something about proteins. You'll recall proteins have a three-dimensional structure, and this three-dimensional structure controls their function. So, for example, supposing the function of the protein is to grab something, and this is the three-dimensional structure it has. It's all set here to grab something. It's sit in this function. If you change that structure in any way by the order of amino acids, it doesn't work. Okay? It's got the same rest of it, but the active part doesn't work. Protein kinases, as shown in the graphic, have the ability to catalyze the addition of a group called phosphate. It's a small organic thing of phosphorus with some oxygen atoms, to proteins. And when you add phosphate to a protein, it changes its structure. So in the same way that if I change the amino acid sequence, I could change the structure, adding phosphate to a protein target changes its structure. In the case of the maturation promoting factor or cell division stimulus, essentially it changes the structure to activate it. So it might be like this, and now you add the phosphate and it opens up, and now it goes ahead and stimulates cell division. The very first topic I discussed in this course, Gleevec, this miracle drug for blocking chronic myelogenous leukemia in the basketball player is a protein kinase inhibitor. By the way, for his work, Hartwell won the Nobel Prize in physiology and medicine. The lesson of this story: cancer is a cell disease. What goes on in cells is very important to the control of cancer. So in this lecture, I'm going to describe, first, cells are the basic structures of living things, what they are. I'm going to describe how human cells have internal structures to carry out various functions, and that human cells can become specialized or cancerous, but in both cases, they retain the entire genome, all the chromosomes, all the DNA in the nucleus. The next lecture, I'll go on to discuss from the properties today, in this lecture, the unusual properties of cancer cells and the fact that they divide without normal controls and the fact that cancer cells don't die. So my first topic is cancer cells and normal cells are basic structures of living things. I want to introduce that in kind of an interesting way and talk about a single disease. The disease is pneumonia. Pneumonia you'll be familiar with, possibly. You end up with chills and pains and cough and a fever, chest pains, and then junk in your throat that you cough up, yellow, greenish gunk. We can call it gunk, we can call it phlegm. The docs will call it sputum, so that's what I'm going to call it, sputum. If you look at the sputum and you look inside, you see very small rods, typically, rod-shaped objects. They're extremely small. They're about 1 micrometer in diameter, which is about 1/20,000th of an inch, 20,000th of an inch. And now you can do an experiment. You can say, does this thing, which happens to be in the sputum, actually cause a disease? You do an experiment. You take the sputum and this rod-shaped thing, you inject it into a mouse, a laboratory animal. You watch and see if the mouse gets pneumonia. Hopefully you'll treat it and get better, but you watch the mouse gets pneumonia, and then you look at the mouse and you see if the mouse has sputum, which it will, and you re-isolate the bacterium. This type of experiment where you inject and recover and get the same symptoms was first derived in the 19th century by a great microbiologist, Robert Koch, and it's a very important series of experiments in looking for the causes of infectious disease. I described this earlier on in the course when I described a bacteria, Helicobacter pylori, that's involved in stomach cancer. Later on, I'll describe a virus that's involved in cervical cancer. The same types of experiments have been done. If you take the sputum, this gunky stuff, and you filter it through a filter, like a coffee filter or something like this, only with smaller pores. The pores here are 1/2 of a micrometer, half the size of a bacteria. You look at the filtrate, the stuff that comes out of the filter, it will, if you inject it into an animal, not cause pneumonia. So this agent that causes pneumonia is larger than 1/2 of a micrometer. What is this agent? It's a bacterium. It's a cell. You can treat it with an antibiotic and get better. You can grow it in a laboratory dish and give it medium, just as goodies that bacteria like, sugars and salts and things. And it'll live. It has complex chemistry. I showed you that the other day. Complex reactions going on. It's a complex chemical thing. It's got thousands of things. It's got DNA, etc. So that's one type of pneumonia. Here's a second type of pneumonia. In the second type of pneumonia, it's caused by a virus. You inject the sputum into the mouse, you get pneumonia, you recover and repeat. And you look and you filter it again through a filter, and the stuff goes through the filter. It's too small. That is, the clear filtrate, in your case would be the coffee, not the grounds. The clear filtrate, you inject it into an animal, it doesn't give the disease. It does give the disease in this case, actually. So this is a much smaller particle. It's about 1/2 millionth of an inch. These are viruses. They only grow inside of cells. They're composed, as you'll find out later on in the course, and I've alluded to earlier on, of proteins and some DNA. It's not a cell. So we got a cell, and we'd have not a cell. The cell theory says, in biology, 3 things. First, the cell is the unit of biological structure and function. Second, it's the unit of life. Life is made of cells, and all life comes from cells, except for the first cell, which came from chemistry. And third, that cells are the unit of biological continuity. And I mentioned before, humans typically have 60 trillion cells. So here's a single fertilized egg in a human, and the single fertilized egg gives rise to a complex human. Human cells have internal structures, and they carry out various functions. I want to introduce those structures because I will come back to them when we talk about cancer cells. We can recognize two types of cells in the living world, complex ones and simple ones, and I'm using these balloons to give you some ideas of internal structures. You know, some places you have just a plain balloon, and the plain balloon can be likened to what's called a prokaryotic cell, a simple cell called like bacteria. It doesn't have a nucleus, doesn't have any special structures inside to speak of. They're highly successful, lots of them out there. In fact, we have 100 times more bacteria in our bodies than we have human cells. So we're all living together. We're not just human cells. The other type of cell is called a eukaryotic cell, like a human cell. It's got balloon substructures. Right? You see these substructures inside the cell. They're much more complex. A major way of studying and diagnosing cancer is to look at cells. Now, cells are too small to see with the naked eye, so you need microscopes to see cells. And so here is two types of microscopes that are used to see cells. One type is called the light microscope. It's the familiar microscope where you see a doctor or person looking through the thing at something on a microscope stage, looking very scientific. That is used overwhelmingly in diagnosis and treatment of cancer. We can use dyes to stain and find out what composition of the cells are. And we can look at what the overall structure of the cells is and how they relate to other cells and tissues. This is what a pathologist does most of the day. The electron microscope sees a lot more detail, requires a lot more preparation time, and is not used that much diagnostically. It's used in research, but it's not used that much diagnostically in terms of cancer. Well, let's take a tour of the cell. Let's look inside a cancer cell and see what we can see. We see a lot of different structures. The outside of the cell, I want to emphasize, is what's called a cell membrane, which is the outside of the cell. We'll discuss it in more detail later on. The inside has a large structure called the cell nucleus. And again, it's one of those balloons inside the cell. And there's a couple of other structures that I'm going to describe. Let's look at these structures in a little more detail, just to introduce them to you. First, the cell nucleus. The cell nucleus contains its DNA, and it's where the expression of DNA to RNA, remember that copy of the blueprint that's going to direct the synthesis of proteins, is made, and it's separated from the rest of the cell. There's good reasons for this separation. It really makes a much more effective cell to have DNA kind of insulated from the rest of the cell from any damage that's going on outside. But it's a hard problem here for cancer. If we're using drugs that we want to target to the cell DNA, and you're going to see a number of drugs do this, there's going to be a problem because it has to get through that nuclear membrane, that is the barrier that contains the DNA, to get to the DNA. So there's intensive study going on of the transport of substances through that nuclear barrier. Here's another part of a cell. It's called the mitochondrion, fancy name. There's a lot of these inside the cell. It provides energy. It's the powerhouse of the cell. So it's where sugars are broken down, etc., to give energy. Of more interest in terms of cancer is the cytoskeleton. Well, it is what it is, right? Cyto means cell. Skeleton is skeleton, right? So it provides a shape inside the cell. And you'll see that cancer cells are changed quite a bit in their cytoskeleton. In addition, it's railroad tracks. These are fibers that are kind of railroad tracks inside the cell. So little cars, molecular cars, can move along the tracks from place to place inside of cells. It is also the place that controls the movement of chromosomes. You've seen, again, several times in the course, including today, that chromosomes in the middle of the cell, and then they migrate to the opposite ends of the cell during cell division. They move along these railroad tracks of the cytoskeleton. Because of this, because of the chromosome movement, this is a target for chemotherapy drugs in cancer. We'll learn about that later in the course. I want to talk a fair amount about the cell membrane, the boundary outside of the cell. The basic structure and function of the membrane is to separate the cell from its environment and to interact with other cells. So first, I'll talk about the barrier. The membrane is a structure. Well, that means that it doesn't dissolve in water. The cell is 75% water. The environment of biology is overwhelmingly water. So when I dive into the pool, I don't dissolve. Now that's maybe trivial, but actually what it means is the substances that make up my membranes and my bones are things that don't dissolve in water. In this case, they're like oily, fatty substances, and those fatty substances provide structure in biology because they don't dissolve in water. We call things that don't dissolve in water hydrophobic. They don't like water. We call things that dissolve in water hydrophilic, which are things that tends to dissolve really well. Problem here again of drug delivery. A drug has to cross this oily, water-hating membrane. And if the drug is something that dissolves well, and you know that, you've seen cancer patients getting intravenous therapy, and there's drugs that are dissolved, they get into the blood system, and now they arrive at the target cell, and now they got to get across this barrier. This barrier is fatty, and it's hard for the drug to get across. So that's a strong consideration for drug design. So the barrier, that's one function of the membrane. Here's another function of the membrane, signaling. I've talked about this in this lecture as well as other lectures. A signal is made outside of the cell. It knocks on the door. Wait a minute, what's it knocking onto? I mentioned this before, and I showed you this in growth factor. Here's a signaling molecule. It's a protein in the membrane. It's sitting in the membrane. It's the binding site for the signal. And then something happens to this after binding to signal things going on inside the cell. So this is highly specific. Different cell types have different membrane receptors, so they will actually respond to different signals. Here's another function of the membrane that's important for cancer. The cells interacting with neighboring cells. Cells are not isolated. Look, I have my skin. All the cells are interacting with each other. And what's interesting is I'm pulling up my skin here. There's bones below it. The skin is not sticking to the bone. That's not a trivial statement, because what that means is the skin cells have something that binds themselves to each other much more strongly than skin can bind to the bone. Those are properties of proteins in the membrane as well. And so proteins on two cells interact, and they will bind to each other. For cancer, this is going to be very important because cancer cells migrate to other places in the body. The cell membrane interacts with stuff outside of the cell. There's an extracellular coating outside of the cell. It's called the ground substance. And this coating is outside of the cell. It's protective. It's composed of things like collagen, which you may have heard of. And there are proteins of the membrane that actually interact with the collagen. So here's the cell coating, and here's the protein that's interacting. So that's going to kind of hold the cells in place, right? So the cells are being held in place by two things, interaction with other cells and interacting with the coating as well. As you will see, for all of the membrane functions I've described, almost all of them are encoded by DNA because they are all properties of proteins. And if you mutate DNA, as has happened in a cancer cell, you will make abnormal proteins, and the abnormal proteins might be something that will alter the properties of the membrane. I want to talk about the specialization of cells. Cells can become specialized, or they can become cancerous, but the important thing to note is they retain the entire genome. The entire DNA is all maintained. Here are tissues. There is a process that we have a fancy name for a cell that's kind of boring, like an egg cell, giving rise to all the different tissues. We call that cell differentiation. You can call it specialization. I'm going to give it its right name, which is cell differentiation. An undifferentiated ordinary cell in the embryo has the capability ultimately to form all the different tissues in the body. And these tissues include things like nervous tissue and bone and muscle, epithelial coating of the skin, blood tissues, etc. Give you an example. The intestine, okay, which is a tube in your digestive system, has at its very surface epithelial cells. These are cells lining the intestine. And if you go below the epithelium, there's a lining of cells called muscle tissue and blood, etc. An important aspect of these specialized cells is that most differentiated tissues, most cells that are specialized, do not divide. They're just doing their thing, and then they die. And they're replaced by other cells that are undifferentiated that then differentiate. So they're non-dividing cells. Now, this is really important because if a non-dividing cell gets its DNA damaged by one of those things we talked about in the last lecture, that DNA damage there's a lot of time to repair it before DNA duplication happens and the DNA damage becomes permanent. So typically, cancers arise in undifferentiated dividing cells much more than in differentiated specialized cells. Now, most tissues have a pool of undifferentiated ordinary cells that are capable of division, so they can replenish the cells that are lost. Again, cells of your skin are constantly being sloughed off, and etc. And you have cells that are unspecialized at any given moment, and they can divide and divide and then specialize to form the differentiated cells. Those are called stem cells. Stem cells, again, are undifferentiated. They undergo rapid division, so if they get mutated by some agent that might cause cancer, there's not enough time to repair, and then you'll get cancer. So the most common cancers are ones where cells are constantly being replaced, like in the epithelium that lines organs, etc. These are called carcinomas. We'll come back to that later on in the course. Cancer cells are different. They look different. Here's the cell that I study. I study small cell lung carcinoma, and you see in the graphic there's a normal lung cell, kind of a flat pancake kind of thing of the pancakes that I make. They usually aren't round. They're kind of not well done. And the cancer cell looks and acts really different. It's not really green and brown. Those are false colors. This is an electron microscope picture of the cancer cell with the normal cells in the background. Differentiated tissues, specialized tissues, are differentiated because they make special proteins to do specialized things. Look, I mentioned before, hair follicles, right? They make the hair protein, keratin. Immature red blood cells make the blood protein, hemoglobin. Cancer cells, they're making cell division kinases, right? Those things that promote cells to divide. All cells have some proteins in common because they got to do cell things, right? Make a nucleus and make protein synthesis and have energy and make mitochondria, those things I showed for energy. So those are housekeeping. So if you look at our genome, we'd say, well, a small part of the genome is housekeeping, just to keep things going and make it a normal cell. But then there are all these specialized functions that act in different cells. When we look at cell differentiation, specialized cells, we can think of two possibilities for the state of the DNA in a specialized cell. We could say, look, in a skin cell, some DNA has been lost. It's only got the DNA just for what it does. Skin, right? So it's going to make skin things, hair and other things, and skin things. And it doesn't have any DNA left for hemoglobin because it's not a blood cell. The other possibility is all the DNA is there, but some of it is turned off. This is important for cancer because if a cancer has mutated DNA and deranged proteins, we're going to try and treat the cell and correct those deranged proteins, but if we can't, we can stop them from acting. We're hoping that all the rest of the cell functions are there so they can take over. How are we going to show whether the nucleus has all of those genes there in a specialized cell, but they're not active? One way to do it is in a nuclear transplant. Let's ask the following question. If we take the nucleus from one cell, it's doing something, and we transplant it into another cell, we remove the cell nucleus of an egg cell, for example, and we transplant a nucleus from a specialized cell in there. If that nucleus had lost a lot of its DNA, it's not going to act like an egg cell nucleus at all. It won't have the ability to. It's lost its DNA. On the other hand, maybe that specialized nucleus has all the DNA. It's just silenced. And if it does, it'll go into an egg and say, hey, you know, the egg cell, its contents will say, form an embryo. Well, this was the basis of one of the most famous experiments of biology recently. A nuclear transplant was done. This graphic shows you how the nuclear transplant is done. It's actually photographs of a nucleus being removed from an egg and a nucleus of a somatic cell, a differentiated cell, being inserted into that egg. The experiment I'm talking about, which proved that all cells have the same DNA, is the experiment that resulted in Dolly the sheep. In this experiment, an egg was removed from a sheep. The nucleus was removed from that egg, and the nucleus from another brand of sheep, another variety of sheep, was inserted into the empty egg. And then the question was asked, can that nucleus prompt an entire lamb to be born? And that's exactly what happened. That's Dolly the sheep. And so this proved that the cell nucleus of a differentiated cell is totipotent. By extension, all cancer cells have all of the DNA of every other cell. And there may be some changes, but if mutations happen and we treat it, we can hopefully allow other functions to take it over. Kind of the epitome of this in a cancer is a teratoma. A teratoma is a rather rare form of cancer. It's 1 or 2% of all cancers. In a teratoma, you've got a ball of cancer cells growing in the abdomen typically. And if you look inside that ball of cells, it's got all types of differentiated stuff going on. It's going to have hair and teeth and bone and even parts of eyes and all sorts of stuff growing in this ball of cells. That says that all the cells in that ball of cells had the ability to form all of the other cells. Summarize this lecture. I described cells as the basic structures of living things. And then I described some of the internal structures of human cells. And then I made the point that all human cells have all of the DNA, and that includes cancer cells have all the DNA. And we can try to manipulate that as we treat cancer. In the next lecture, I'm going to describe how cancer cells have unusual properties with all the things I've described. And that'll form a basis for going on and describing how tumors grow later on in the course. Thank you.